using sugars as Haworth projections is really, really convenient, but it's not actually very accurate, because we know that sugars are not actually, or ring structures like that, are not actually planar. And in fact, we can look at some of the conformations that you tend to see on either a furanose or a pyranose ring. I have here built with some of my models a furanose ring, right, because it's a 5-membered ring. And if we find then the oxygen, and based on that locate a carbon number 1 and carbon number 2, one of the structures that can occur in a furanose ring is actually something termed a C2 envelope. So what that means is that all of the other carbons besides carbon number 2 are within the plane of the ring, but carbon number 2 is outside of the plane of the ring, so it makes something that looks like an envelope, where if I were going to, lick this envelope and seal it, I'd look like that, right? And the part that carbon 2 is the point on, and then you would fold it over like that. Oops, kind of broke it. But that gives you an idea of the C2 envelope, where only carbon number 2 is actually outside of the plane of the ring. Another thing that actually can occur is when you have a carbon 3 envelope, so you can actually see a C3 envelope as well, so where instead of carbon 2 being out of the plane of the ring, it would be carbon 3. So there now we have only carbon 3 out of the plane of the ring, and the others are sitting within the plane. Okay. You could also get something termed a twist. And in a twist, you actually see that carbons 2 and 3 are both outside of the plane of the ring. So in a twist, carbons 2 and 3 both are outside of the plane of the ring. So you can kind of see there then that kind of juts up, and it does look kind of twisty. Okay, so carbons 2 and 3 both outside. Okay, so those are some different conformations that we tend to see with a furanose ring, so a 5-membered ring. Let's go ahead and look at a pyranose ring. So here's a pyranose ring, 6-membered, right? Actually, no matter how I manipulate it, it really doesn't quite look right flat on, because it doesn't sit in a plane. So we get 2 potential conformations for a pyranose ring. I think you guys are familiar with both of these. So the 2 conformations that we tend to see are 1, a boat, and 2, see if I can get this right, a chair. Okay, so the boat and the chair conformations are the common conformations for pyranose rings. Look at it. It looks like a Barco lounger. I mean, you could just plop right down, ball right there. Okay, so this is the chair, and the chair actually tends to be the most favored structure because it actually puts the grooves in a position where steric hindrance is minimized. And particularly, now there are two positions off of each carbon in a chair conformation. You can get what we term the axial position, which is sort of up and down and perpendicular to the chair. That would be where you'd put your beer if you had one of those Barker loungers with the beer container. Those are so tacky. But in any case, that's the axial position. Now the equatorial position, that one right there, now that one's going to shoot away from the ring. So the most sterically favored conformation is to put the big bulky grooves in the equatorial position. Derivatives of sugars play extremely important roles in biological systems. Let's take a look at what some of these derivatives are and draw examples. The sugar phosphates are very, very popular, very important molecules. And in fact, you might guess that this means that they are phosphorylated sugars. So let's begin with our very famous, very favorite glucose molecule. And consider glucose 6-phosphate, a very famous phosphorylated derivative of glucose. So CH2OPO32-. This is a a sugar phosphate, and very important when cells are bringing in glucose. Oftentimes they will phosphorylate it so that it no longer counts against their concentration gradient, and that allows them to concentrate it. Let's take a look then at sugar alcohols. A very famous sugar alcohol is glycerol. So if we modify the aldehydic carbon of glyceraldehyde that we have already talked about, and it is instead converted to an alcohol. So let's erase here carbon 1, and if that is an alcohol, this is glycerol, so sugar alcohols. We can also get sugar acids. You might guess that's going to be carboxylic acids. So there are two types. There are aldonic acids, as well as algironic acids. Let me show you an example of an aldonic acid where carbon 1 is converted from an aldehyde to a carboxylic acid. So we might look at this example. Gluconic acid. And in fact, gluconic acid can cyclize and literally form a lactone, and vitamin C is a lactone. So algironic acids, this is where the highest numbered carbon, instead of carbon 1, the highest numbered carbon, so in glucose that would be carbon 6, becomes a carboxylic acid. So I'll let you practice writing that. Let's take a look at deoxy sugars. So these are actually extremely famous. In fact, as I say, deoxy sugars, you might be thinking deoxyribonucleic acid, DNA. So deoxyribose is a very important component of DNA. And look here at the second carbon. 2' carbon in DNA, number 2 carbon here in the sugar itself, it is a deoxy. So there is no hydroxyl there, whereas in carbon 3 there is, and of course in ribose itself, there is. So deoxyribose. And then amino sugars, maybe one of my favorite. Well, let's just play with our good friend glucose and create glucosamine. At carbon 2, there's an amino. So this is glucosamine. We can also get N-acetyl glucosamine, so where we literally see a situation where an acyl group, and specifically an acetyl group, is added onto the nitrogen. That's a double bond O. N-acetylglucosamine. Oh, this is starting to maybe ring some bells for those of you who studied some microbiology. N-acetylglucosamine, we termed it NAG, very, very important part of the bacterial cell wall. So big role that these play in biological life, and we'll see more of them.